Hello, everyone. Welcome to MD 6028. This is the first lecture of the module. What I really want to do in this lecture is introduce some of the key concepts you need to understand. So it's kind of a foundational lecture where we cover we give you a strong foundation that the later lectures will build upon. And I also want to talk about some general things kind of around the module, explain the way the module's going to be delivered and things like that. There's a separate presentation that's going up alongside this one that will be an introduction to the technical aspects of the module. So that will be about timetable, it will be about assessments, feedback, it'll be about things like that. This module is just going to be a quick overview of the delivery method and an explanation of why the module is being delivered in the way it is, and then we're going to delve into the actual scientific academic content in a bit more detail. And again, we're going to then establish those fundamental concepts in virology that we need to understand straight away, essentially. So if you've seen the announcement I put on the module space, you would be expecting this method of delivery, a recorded lecture that you can watch in your own time. I've made the decision to run the module like this for the first month, and then we can kind of reassess and see how things, see how the delivery method is received, and see whether we want to keep going forward. If it's not working, I'm absolutely happy to change and just go back to a live delivery method. The problem with the live delivery method I see on this module is we have these 3-hour slots. 3-hour slots can be good because it means you have this big gap at the end of the year where you don't have taught sessions for this module. And in that gap, you can focus on exam preparation, you can focus on dissertation write-up and things like that. So there's benefits to it. However, the downside is 3 hours is a long time to sit and listen. It would have worked in person. When the module was originally planned, it was going to be a face-to-face module, and it was going to be a lecture for, say, 50 minutes, then a 10-minute break, then a lecture for 50 minutes, then a 10-minute break, then an hour-long tutorial. And that kind of works. It works quite well face-to-face. It doesn't really work on Teams because it's just too long to be sitting in front of a computer. Even though the tutorial is interactive, it's slightly different to how it would be in person. So I just think it would be a struggle to go through a full 3-hour session and expect everyone to be attentive, expect everyone to get the most out of the material. So I think using this method of delivery where I pre-record the delivered lecture content and then you're able to watch it kind of at your leisure whenever you have free time, I think there's real benefits to this. Again, we'll assess in a month and see if it's working or if it's not. I understand watching recorded lectures isn't the same as listening to a live lecture. It's not quite as engaging because you can't ask questions, but I'm hoping in the tutorial sessions that's when you'll be able to ask the questions. So you'll be watching the recorded lecture in your own time whenever you can. Then you'll be coming to the tutorial sessions with questions prepared. And in those tutorial sessions, what I'll essentially be doing is asking you to explain concepts from the lecture back to me, and then we'll explore those concepts. So that's why you really do need to watch these lectures before you come to the tutorial sessions. And this has a potential to be a very, very good way to learn because regardless of the environment or mode of delivery, just me explaining something to you is not the best way to learn something, because there's it's not the knowledge isn't being explored by you. You're not exploring the concepts, you're just listening. But when we have these interactive tutorials, it's an opportunity for you to really explore the knowledge and apply it and kind of pull it apart a little bit and make sure you really, really do understand it. we call it active learning. So you're actively taking part in the learning process. You're not just sitting back and listening. And obviously sitting back and listening is really useful in the right context, but we can really strengthen your learning by having this active participation element. So in terms of your timetable, again, this is mentioned on the module space. So if this is confusing, have a look at how I've written it up on the module space. If it's still confusing, just let me know, send me an email or give me a Teams call, that's fine. But the way it's going to run is each 3-hour scheduled session in your module, you're only going to be required to actively attend a session for the last hour. The first 2 hours are basically a gap where if you want to watch the pre-recorded lectures, you can watch them then. If you don't want to watch them in that time, you can watch them the day before or 2 days before. That's absolutely fine. But that 2-hour gap basically gives you the opportunity to watch the pre-recorded lectures. The final hour of the timetabled slot, that is an active participatory tutorial that we'll be doing through the Teams channel for the module. So for the first week, the sessions are scheduled 9:30 to 12:30. So between 9:30 and 11:30, that's your own time. Again, potentially an opportunity to watch the recorded lectures if you haven't watched them the day before or the day before that. But 11:30 to 12:30, I am asking you to come on the Teams channel and take part in the active learning. In terms of attendance, the attendance is going to be monitored as it is with any other type of lecture or tutorial. So we are going to be expecting you to attend and sign in, and if you don't, you're going to be getting emails and things like that. So they are compulsory sessions. So I do, I do expect to see you there and hope to see you there. If there's reasons why you can't be there, just as the case with other lectures, if you're ill or something like that, that's absolutely fine. Just make sure you report your absence and go through the proper channels. What we don't want is to see students dropping off. One thing we're very conscious of in this kind of online phase of learning is just students slipping through the net, students slipping away. If you don't physically see a student and you physically see them missing off the register, it's noticeable because it's in person. But on Teams, things are slightly different now, and I'm worried, and a few other members of staff are worried it'll be easier for students to just slip under the radar, and then what happens is students fall more and more behind. I don't predict this happening for any of you, particularly as you're on my program, so I'll be I keep an eye on your attendance through that as well. It's just something to bear in mind. So hopefully that makes sense. Okay, so I think I've covered everything I wanted to say there. So we'll go on to the content. So the scientific content, what are viruses? This module is called MD6028 Virology. So there's going to be a huge focus on viruses. That's what we're talking about. As I'm sure you know, ology just means the study of something, zoology, study of animals, psychology, study of the mind, virology, study of viruses. So the first thing we need to establish and the first thing we need to understand is what viruses are. And we can answer that question in a number of different ways. One way in which we can answer that question is from a physical perspective. So if we're just thinking from the point of view of what physically is a virus, and the most obvious answer in terms of what physically is a virus is it's something that's very, very small. That's one of the most defining physical characteristics of a virus. It's tiny. We know it's microscopic. That's why we talk about it in microbiology. Microbiology is, again, if we break the word down, ology is study, bio is life, micro is small. So microbiology is the study of small forms of life. Specifically, micro refers to anything that's too small to view with the naked eye. So microorganisms are generally too small to view with the naked eye. That's why they're called microscopic. This includes viruses. However, even for microscopic organisms, viruses are small. So we have some points of comparison coming up on the next slide. And the observation that we want to make is that viruses are not only much smaller than human cells, so much smaller than eukaryotic cells, but they're also generally much smaller than bacteria. So here we have E. coli, which is a bacteria, so it's just a single cell there, and 0.5 micrometers. And here we have the diameters of a few different viruses, and you'll see the diameter for HIV is 0.12 micrometers, 0.04 micrometers for HPV, and smallpox is 0.2. So again, generally speaking, we're seeing the viruses are smaller than the bacteria here, E. coli. So we can say in general, bacteria are larger than viruses, but that's only in general. So here, as we said, we have E. coli where the radius is 0.5 micrometers. That's specific to E. coli. So different bacteria are different sizes. And even though viruses are generally smaller than bacteria, there is an overlap. And what that means is the largest viruses are bigger than the smallest bacteria. So it's just a general rule. If you map out all viruses by their size and all bacteria by the size, you'll see viruses tend to be smaller, bacteria tend to be bigger, but there is a significant overlap, and it's not just a couple of species, it is a significant overlap. If you need an example of a large virus that is bigger than some bacteria, we talk about the mimivirus family. So the mimiviruses are some of the largest viruses we know about. They're still viruses because, as we talk about in some later lectures, in some later slides, the way we define viruses, it's not all about size. Size is just one aspect, so it doesn't matter that these viruses are bigger than bacteria. They're still viruses because they still have all the other defining features that viruses have. So just don't be confused if when you're reading up on viruses, you notice that they're bigger than some bacteria. Absolutely fine. We know that some viruses are bigger than the smaller bacteria. Just a note as well on the scales we're using. So on the units, you'll notice I'm talking about micrometers, and essentially what we have here is an explanation of what a micrometer is. So you can think of a micrometer as 1/1000th of a millimeter. So you take a millimeter, divide it by 1,000. Each one of those divisions is equal to 1 micrometer. That means something that is 100 micrometers is 0.1 millimeters. We also have a unit called nanometers. Nanometers is even smaller. So what micrometers are to millimeters, nanometers are to micrometers. So just like 1,000 micrometers equals 1 millimeter, 1,000 nanometers equals 1 micrometer. This is important because when we're talking about viruses, we usually talk in terms of nanometers, and I'll explain the reason for that now. If you look at the bottom, you'll see if we switch from micrometers to nanometers, the values are different. And what you'll see is we've stopped using values like 0.2, 0.04, 0. We don't tend to like using those values. We want whole numbers. If we're using these kind of 0.2, 0.04, 0.12, things are potentially getting messy because what's happening is we're operating at the very edge of the scale. So we're talking in micrometers, and we're talking, say for HPV, we're talking 0.04 micrometers. that's right at the edge of the scale. Why are we using micrometers if we only have 0.04 of a micrometer? It doesn't really make sense. So what we do is we kind of shift down, almost like shifting gears. We go down to nanometers, and then when we express that value in nanometers, we say 40 nanometers. That's a nice whole number, makes a lot more sense. It's a lot easier to visualize what it is. It's a lot easier to compare, even just in our head, 40 nanometers to 200 nanometers. So that comparison is easier than the comparison of 0.04 micrometers to 0.20 micrometers. These numbers are just easier to work with. So again, that's a good general rule within microbiology and biology actually. We don't want to be operating on the very edge of scales. It's just not where we want to be. If you think about using a pipette, we're essentially applying the same principle. When you're picking which pipette tip to use, you don't want to be using, you know, a 1,000 microliter pipette to be dealing with small volumes like 1 microliter or 2 microliter. You want to be, because you don't want to be operating at the edge of the pipette's capacity. You don't want to be operating at the extremes. So it's a similar kind of idea here. As you can see for E. coli is 500 nanometers, which again is the same as 0.5 micrometers. And this kind of explains why when we're talking about bacteria, generally we're going to use micrometers, because 500, it's quite a high number. It's getting up there, and again we want to avoid using the edges of scales in both directions. So 0.5 micrometers, that is the end of the scale the other way, but it's nothing like 0.04. So yeah, generally for bacteria, we won't use nanometers because we just end up with a whole bunch of zeros on the end, which can get a little bit messy. So the point I've made there in the last few slides is that viruses can be physically defined as being small and generally smaller than bacteria, but not always. So it's not an absolute rule, but generally viruses are smaller than bacteria. Okay, so we can also define viruses biologically. So if we think from a biological perspective, what is the biological composition of a virus? So what biological components make up a virus? I'm sure we can all answer that question for a cell, so for a living cell. So if we think of one of our own cells, a eukaryotic cell, if we're talking about the biological components, we can say there's protein, there's a nucleus, there's a plasma membrane, there's all the other organelles, there's DNA, there's cytoplasm with a whole bunch of protein floating around in it and enzymes. I'm sure all this sounds familiar to you. That's the biological composition of one of our cells. And if you want to talk about the biological composition of our cells overall as an organism, as a macroorganism, so macro meaning big, visible to the eye, we can say that we're a composite of, you know, this huge number of different cells, and each of these cells have the biological composition that I've mentioned. For viruses, the situation is very, very different. Viruses are acellular, so they're not composed of the same components that constitute a cell. And that's true for a eukaryotic cell, for one of our cells, or a bacterial cell, a prokaryotic cell. So viruses are different from both those forms of life. And that principal difference is that viruses are not cellular. They don't have the components that make up a cell. The components they do have are listed here. And when the example given here is HIV, and what you'll see is this kind of tan circle around the edge. This is a lipid envelope. The lipid envelope is not a cell membrane. Don't make that mistake. They're not the same thing. they're composed of similar things. They're composed of phospholipids, but the reason they're composed of phospholipids is most viruses will obtain their lipid envelope from a cell. So viruses, when they're infecting a cell, they'll produce more copies of themselves, and then these copies will leave the cell. And as they leave, they'll steal a little bit of the cell membrane, and this cell membrane then becomes the viral envelope. So we'll talk a bit later about the specific distinctions between the viral envelope and the cell membrane, but it's really important we remember they're not the same thing. We can't talk about the virus cell membrane because there's no such thing as a virus cell. You'll notice there's also proteins studded in this lipid envelope. Most viruses will have proteins studded in their lipid envelope, and these proteins serve a range of roles, often serving a purpose in the attachment to host cells. So they're often functional in infection. When a virus infects a cell, it has to attach to it first, right? And often these proteins that are embedded in the lipid envelope mediate that attachment. It's worth noting that not all viruses have this lipid envelope. So one of the ways we can classify viruses, and we'll talk about this later, is whether or not they have a lipid envelope. And the reason we can do that is because a lot of viruses don't. Whether or not a virus has an envelope has really significant implications on the way it replicates, on the way it infects cells, and all these kind of things. So it is really, really important, and we are going to talk about that more at a later date. But for now, we just need to know that some viruses have this lipid envelope, some don't, and those that do often have proteins kind of embedded in this envelope in a way that means they're kind of studded on the outside. Within the lipid envelope, you'll see here that we have something called a matrix. This is a HIV virus particle, and HIV does have this matrix, which is just basically a mesh of different things holding everything in place. But the matrix isn't a particularly important part, and certainly not all viruses have it. The next important component is actually the protein capsid here. So you'll see here this almost bell-shaped structure. This is the protein capsid, and you can essentially think of the protein capsid as a shell. And this is a protein capsid that encapsulates the genome of the virus. So within this capsid, you can see the viral genome is present. And alongside the viral genome, there's often some other proteins floating about. Functional proteins often involved in infection or replication. What you'll notice is not present is a nucleus or a nucleolus. So nucleus is what we have in eukaryotic cells, as is nucleolus, and then in prokaryotic cells we use the term nucleoid. You'll notice they're both absent here. The area where the genome is present, not a nucleus, not a nucleolus, not a nucleoid, it's just the internal or the inside of the capsid. So again, you have the capsid, protein shell, genome in the middle, and then outside this capsid shell you sometimes have this lipid envelope. Some other things you might notice are missing: no mitochondria, no Golgi apparatus, no tRNAs, no cytoplasm. We've said no cell membrane, obviously no cell wall, which you see in some cell types, not ours, but some cell types. So again, it's pretty clear that the virus particle, not cell, is not a cell organized in the same way that a cell is. It doesn't have the components that constitute a cell. And the simplest way we can summarize the biological composition of a virus particle is by saying it is a nucleic acid genome enclosed within a protein coat, which may or may not be surrounded by a lipid envelope. That's quite a neat little summary there. I don't know if I wrote that. I might have got it. I don't know. There is a good quote describing viruses, which I definitely didn't write, but it goes, a virus is just a piece of bad news wrapped up in protein, which is essentially true. So if you think about the minimum composition that you can have for a virus, I've told you not all viruses have a lipid envelope. So a lot of viruses will just be the genome wrapped in a protein shell. And we can correctly describe that genome as bad news, because once that genome gets inside your cells, if the virus is evolved to infect humans, then it's potentially going to start infecting your cell. And we know a lot of different viruses do infect humans. A lot of them we're going to talk about in this module. A lot of them we've talked about already in different modules. So yeah, I think that's a neat way of describing a virus. And what we have here are some representations of different virus particles. What you'll notice is that these are hugely varied in their shape. We have variola at the top, so smallpox family, herpes virus next up, then we have this bullet-shaped virus, which is rabies, and then we have a lot of spherical, but they're not really spherical, but that kind of shape. Some of them are icosahedral. Then we have Ebola virus, which has this long, almost worm-like shape. So quite a lot of variation in terms of how they look. However, if you break down the constitution of each of these virus particles, you'll see that they meet the criteria we lined out in the last slide. So you'll see that each of these viruses, despite their differing shapes, they are composed of a genome wrapped in protein, and that may or may not be wrapped in a lipid envelope. So that's why we need to remember that definition, because we're looking at all these different viruses that all look very, very different. And we need to remember what makes a virus. So what do these things have in common? What do these things that look so different share? What are the defining features? What makes a virus? And one of the things is this biological composition that we discussed in the last slide. So there's an important consequence of the minimalistic composition of viruses. So if we can compare the composition that we just talked about, so what makes up a virus, to what makes up a cell, we essentially said that a lot of things are missing. So the nucleus is missing, all these organelles are missing. Obviously they're not missing, but if we're doing a comparison across, we see they are in cells, but they're not in viruses. So how on earth do viruses cope without all these fundamental structures, without all these how do they fulfill the basic processes of life if they don't have these structures and these elements? The answer is they don't, at least they don't independently. So because they're so minimalistic, because they lack all these organelles and other structures, viruses are completely incapable of fulfilling the processes that define life independently. So there's this big debate that's been going on since viruses were first discovered as to whether or not viruses are alive. If you have a virus on its own, it doesn't do anything at all. It can't reproduce. It can't produce energy. It can't respire. Can't grow, can't transcribe, translate. It's what we call inert. It's inert. How can it translate and transcribe if it doesn't have the machinery required to do that? how can it produce energy if it doesn't have mitochondria, it doesn't have chloroplasts, doesn't have any of these structures that it could potentially use to produce energy? It can't. So again, we define viruses as inert when they are on their own. However, when they infect an individual, viruses enter into cells. So viruses enter into the intracellular environment of the host. And the reason viruses enter into the intracellular environment is because they need access to all those structures, to all those enzymes that we talked about, all those structures that we said viruses don't have. Viruses will enter into host cell and use the structures that are already there. So they will manipulate host cell machinery in a way that enables them to replicate and transcribe and translate and do all these different things that enable new copies of the virus to be made. And that's where the debate about whether or not viruses are alive comes from. Because if you have these, so you have viruses on their own and they're dead, they're inert, they can't do anything. They can only do anything at all when they're inside a host. So the line is blurred there. Can we really consider a virus alive if it's completely inert when it's on its own? I ask students this quite often, and usually it's a 50-50 split, and then we explore it a bit more, and some people switch sides, and there's good arguments both ways. This is something we'll talk about in the tutorial session. So if you have any thoughts, please do prepare them. If you have any outside reading that you think would inform this discussion, please do look up some papers. It's a really interesting discussion, and it's one of those areas that is unsolved, that no one can really agree on, which is good because it's quite exciting to talk about. It's one of the good things about being at level 6 as well, because you're better placed to explore these concepts and ideas, and it's not as much about just learning information as it was at level 4 and level 5 as well for a lot of it. It's a bit more about exploring these ideas. So if you can think about that, that would be great. The predominant feeling amongst a lot of kind of experts and academics is that, and I kind of agree, is that the most useful way to think of this is you think of viruses as having a dead stage and an alive stage. So when they're outside the body, they're dead, they're inert. Again, we avoid saying dead because dead has it evokes certain philosophical implications, and it's dead means a lot more than what we intend it to mean in this context. So what we can better describe a virus as is inert. It's not doing anything that makes it alive. It's inactive. So it has the inert stage, or again, dead stage, but I shouldn't really say that. And then it has the living stage or the replicating stage. And the replicating stage takes place in the host cell environment. So that's my take on it. Feel free to disagree. As I said, there are some really good arguments both ways. Okay, so this is essentially what I've just talked through. Yes, so this is this is the basis of viral evolution, essentially. The ultimate selective pressure upon viruses is to produce progeny. That's how evolution works. If something enables the production of progeny, it's selected for, and the viral strategy of progeny production that has been selected for and that has been refined is the ability to infect a host. We'll talk about in later lectures why this has happened, and we'll talk about the evolution. Why is why the right word to me? We'll talk about how it's happened. Would be a better way to phrase it. We'll talk about the evolutionary pathway that viruses have followed and how they've ended up in such a strange position. Something I missed out on the last slide was this idea of obligate parasitism. So you'll often see viruses referred to as obligate parasites. That essentially means, or it's a fancy way of saying, they can only replicate if they're infecting a host. So if they're parasitizing a host. Strictly speaking, when a virus infects you, the virus is acting as a parasite and you are the host. And again, that's the only way a virus can replicate. So it's not just a parasite, it's an obligate parasite because it is obligated to engage in parasitism to produce progeny. If it doesn't engage in parasitism, it doesn't produce progeny. So it's an obligate parasite. Okay, here I have some of the different processes that I referred to. So if we think about the processes that define life, we can say the ability to transcribe and translate proteins, the ability to make genome copies, which is replication. So transcribing and translating proteins is genome expression, making genome copies is genome replication, the ability to grow, essentially to assemble proteins and assemble other structures to make a bigger virus particle, or in the next line to make additional virus particles. It's just not able to do this because these efforts are obviously linked to gene expression and genome replication. Generating energy is a big one. To do any of these things, there's an energy requirement. That's why our cells have these complex pathways by which energy is produced. Viruses don't have these, so they haven't got a hope of doing any of these other things because they just haven't got the energy. I mean that in a scientific way. They just haven't got a means of energy production. So another reason why they are obligated to parasitize host cells. Okay, so the next way we'll define viruses is with regards to human health. So if we think about in the context of human health and disease, what are viruses? Another way we can word that is what role do viruses play in human health and disease? And this is linked to the last few slides where I defined viruses as obligate parasites. So the answer is viruses use us as hosts. They engage in a relationship with us in which they are the parasite and we are the host. What that relationship means is the virus benefits whilst we lose out. And that's what a host-parasite relationship looks like. Host loses, parasite wins. So what does the virus win? The virus wins everything we talked about in the last few slides. The virus gains access to our cells and it gains the ability to express its genome, to replicate its genome. It gains access to the energy. It gains access to amino acids, which allow it to replicate its genome, all these kind of things, all these things that it couldn't do when it's on its own, it gains the ability to do when it successfully infects a human. And again, I'm just using humans as an example because we're talking about human health and disease, but there are a whole range of viruses that are capable of infecting a whole range of different organisms. Just talking about humans because that's really our main area of concern when we're studying life sciences. We care a lot about human health and disease. We care a lot about the specific diseases that viruses are implicated in. Not that I really have to explain this at the moment with global pandemic, but obviously the viruses that cause human disease they're a problem. They cause a huge global burden. So they're of interest. Again, there's academic interest in all these other viruses that infect other species, particularly the ones that could jump to humans, which is something we'll talk about at length later, the process of zoonotic transmission. So viruses that infect animals that then jump across to humans and become capable of infecting humans. that's a hugely important process. So we do need to talk about that later, but the main focus is going to be for this lecture is going to be the viruses that cause disease in humans. And when we have viruses that cause disease in humans, as we said, they're engaging in a host-parasite relationship with us. And this is what that looks like. We have this slide here, and where we have the virus enters into the host organism, then it enters into host cells, and this is crucial. And this is a big difference between viruses and bacteria. Bacteria don't usually enter into host cells. Some do, some definitely do, but as a general rule, it's not required for bacteria to enter into host cells unless they've involved a very specific life cycle which requires them to enter into host cells. Viruses, however, are intracellular parasites, so they do need to enter into host cells. And again, the reason they need to enter into host cells is because all the processes they need to undergo, the ones we talked about, transcription, translation, replication, etc., these take place within cells, so they take place in the intracellular environment. So if the virus if the virus has evolved to manipulate these systems, to co-opt our machinery, and to engage in these processes, it has to access the internal environment of the cell, because that's where these things are. That's where tRNAs are. That's where the mitochondria is. That's where the ribosomes are. That's where all the cellular machinery is that the virus needs to use. So not only are viruses obligate parasites, they're intracellular parasites. The cell type they enter differs hugely across the different viruses. If we think about the common cold, it's going to enter into epithelial cells of the respiratory tract. If we think about HIV, HIV is going to enter into cells of the immune system. Very different viruses replicating in a very different way, entering very different cell types, but nonetheless, both are still entering into cells because it's a requirement for viruses to do that. Okay, so once in a cell, they will complete their replication cycle. So they will undergo what we call the viral replication cycle, which occurs inside the cell. And again, the purpose of this viral replication cycle is to complete the processes that we talked about, genome expression and replication, etc. And once this process has been completed, the new virus particles that have been produced will then be released from the cell. Once they're released from the cell, they will eventually either infect. Those released virus particles will then infect additional cells within the same host organism, or alternatively, they will infect a new host organism, and that's just a product of chance. Where do these virus particles end up? That depends on a whole host of things that we'll later talk about. Obviously, a lot of these virus particles won't infect anything. They just won't come into contact with host cells that they need to infect, or they just won't come into contact with another host organism. Sorry, I hope that didn't make a big noise. So if you think about when you have common cold, when you cough or you sneeze, there's huge amounts of virus being thrown out in the mucus, in droplets of moisture. And if you're, say, at home on your own and you're ill, these virus particles that are trapped in the mucus or the moisture are not coming into contact with anyone else. They're just getting caught up in a tissue and then thrown in the bin. So these virus particles aren't necessarily going to infect anyone else, but often they will. And that's kind of the viral strategy of replication. Viral replication results in huge numbers of virus particles being produced. And these huge numbers, the idea is that some will infect a new host. And it works. It's an evolutionary strategy that has worked. This production of huge numbers of new virus particles also allows for very, very rapid, the generation of very rapid diversity. So you get these very, very high mutation rates, and that's one of another characteristic of viruses is you see this very, very rapid mutation because they're producing thousands and thousands and thousands and thousands of copies within each. Sometimes within each single infected host cell, you can get thousands and thousands of virus particles being produced. And if you think about an infected host, it's not going to be a single cell that's infected. If you have common cold, you don't just have one epithelial cell infected, you have a whole bunch of epithelial cells infected, and each one of those infected cells is just throwing out virus particles. So you're getting these huge numbers of virus particles being produced. If you just think about random mutations, so chance mutations, there's going to be a lot of diversity in those virus particles produced very, very quickly. And in this slide, I have the common cold virus example I've been using. Specifically, we've called it rhinoviridae here. So a large number of common colds are caused by members of the rhinoviridae family. If you ever see the ending, so the suffix viridae, you know we're talking about a family of viruses. So Retroviridae is the retrovirus family. Filoviridae is another. Again, viridae. So if you see that suffix, it's a family. We'll talk about that in more detail though. So again, most common colds are caused by rhinoviridae. Not all. Adenoviruses can cause common cold as well, so adenoviridae. But what we have here is the generic cycle for rhinoviridae when it causes the common cold. So it enters the host organism through the respiratory system, so through the mouth and nose. Then it enters into host cells, epithelial cells, and then once it's in the epithelial cells, it completes its replication cycle. And by completing this replication cycle, it's producing huge numbers of copies of itself. So it's producing huge numbers of progeny. These progeny are released from the infected cells. And these progeny will either infect nearby cells, so infect other epithelial cells, or they will be passed from the host via mucus and just drops of saliva, and then pass out into the environment as the host is sneezing or coughing or even just breathing. And then once in the environment, there is the possibility for them to come into contact with a new host, and then that new host has caught a cold. Bad luck for that person, but good luck for the virus, because then the virus can do exactly the same thing again and then keep spreading and spreading and spreading. And that is the basis of the miserable cycle of the existence of viruses, just spreading from host to host to host to host, causing quite a bit of misery on the way. HIV is significantly worse, most people would agree, in terms of the consequences of infection. HIV is actually a good opportunity for us to introduce the idea that when we're thinking about the consequences of infection, we often have to go beyond just thinking about the physical consequences of infection. So as you may know, HIV infection can usually be controlled with a drug regimen. So if you give someone the right antiretrovirals and you ensure they can access those antiretrovirals and you ensure they are taking those antiretrovirals, you can control their infection. So in that situation, the physical symptoms of infection are minimal, even negligible, even absent if you're controlling the infection well enough. However, the psychological impact of infection is still there. When we're thinking about these infectious diseases, we have to think about the consequences in a very rounded way. It's not enough to just think about the physical consequences of infection. If we're just thinking about the physical consequences of infection, there's an argument that the common cold is less desirable than human immunodeficiency virus infection, which the vast majority of people would disagree with, because they understand that there is a psychological component to infection, and that there is a very significant psychological burden that's placed upon individuals who are become infected with not just HIV, but other viral infections that are, and here's the key word, that are stigmatized. So there's a significant amount of stigmatization against HIV, against HIV infected persons, and this is a very complicated issue, because we don't want to minimize the consequences of HIV infection, but we don't want to stigmatize people who have HIV. So there's walking along this thin kind of line, if you like, of not stigmatizing people who have HIV, but also being very clear that HIV is a very serious illness, and that people do need to take the appropriate precautions to protect themselves against it. And it's a thin line to walk. Generally, it's done appropriately, and generally people take HIV very seriously, and are happy to engage in practices that protect themselves from HIV, whilst also not needlessly stigmatizing people who have HIV. And again, the key word there is stigmatization, and it's not just about HIV, it's about other infectious diseases. And the stigmatization is rooted in individuals not wanting to contract an infectious disease themselves. Usually, historically, if we think about leper colonies, leprosy is caused by a bacteria, by Mycobacterium leprae. But the principle is the same. People were stigmatized because they had leprosy, and the reason for the stigmatization was because other people didn't want to contract it. So they did things like put people with leprosy in leper colonies. So the consequences of stigmatization can be very real. Nowadays, the stigmatization is quite heavy against viral infections that are chronic, so viral infections that are incurable, because it just adds weight. It adds weight to the infection. The fact that an infectious disease can't be cured, people feel the consequences of that. So if we think about some others, well, we'll talk about these in later lectures anyway, but HSV is one, herpes simplex virus, obviously a lot less stigmatized than HIV. But the principles there, the underlying reasons for stigmatization are there. Also, when infectious diseases are sexually transmitted, there's an extra layer of stigma there as well. The reason why it tends to be viruses is because there's a number of these viral infections that are sexually transmitted and are chronic, or even incurable. Again, stigmatization's a complex thing, and people should be encouraged to take the appropriate measures to protect themselves. But we, kind of as scientists and students of science, what we need to do is be aware of the psychological burden that's associated with these infectious diseases. And again, just be aware of the fact that the consequences of infection go beyond physical symptoms. Okay, so back to the HIV life cycle. So what we have here is the life cycle outlined, and what you'll see is the key stages are the same as they were for the rhinoviruses, same as they were for rhinoviridae. The details are different, the specifics of how each stage is completed are different, but the key principles are the same. So you'll see, just like rhinoviruses, HIV needs to enter a host, then it needs to enter the right cell type, then it replicates within the cell, then huge numbers of virus particles are produced, then these particles will go on to kind of drift off, and some will infect new cells and some will find their way to a new host, and infection will continue. So I'd encourage you to compare this slide to the last slide and see the similarities and see the differences. If we think about the differences, we see that HIV enters new host not through the respiratory tract, but through wounds and micro tears. So wounds, we're just talking about abrasions or maybe needle stick injury, anything that pierces the skin and provides the virus with the opportunity to cross the epithelial barrier. Once it's made its way into the bloodstream, it enters into circulating immune cells. Again, different from the rhinoviruses, which enter into epithelial cells of the respiratory tract. HIV will then complete its replication cycle in a very similar way to how rhinoviruses do, just in different cell type. Then new virus particles will be passed out from the infected cell. These will be released into the bloodstream, and then these will either make their way into another individual through the blood or through the semen, or they will potentially come into contact with other immune cells in the blood, and they'll infect those immune cells. And this allows for the cycle of replication to continue and continue and continue. And this is essentially the life cycle of these viruses. So we've spent some time defining what viruses are, and hopefully now I think you have a clear idea of when we say virus, what do we mean by a virus? What is a virus? That should be clear. If not, we'll talk about it in the tutorial as well. But we need to answer the question now, where are viruses? So these viruses we've been talking about, where are they? Simple answer is everywhere. I've put some statistics and facts on this slide. I made this a while ago. I don't really do this anymore. It seems it's just a bunch of huge numbers that, it's like the point is that there's a huge number of viruses and that they are everywhere. Yeah, that's it. And there's consequences for this. It means we're constantly coming into contact with these viruses. And we'll see that when we're talking about the immune system in this module and medical microbiology. We'll see that the presence of viruses has had huge consequences in that it's shaped our immune system. So you can really see that our immune system has evolved in an environment where we're constantly being exposed to viruses. You can see this. It's a form of co-evolution. We've evolved a kind of balance where our immune system's evolving and viruses are evolving to overcome our immune system. Then our immune system is evolving to overcome viruses or to prevent viruses from infecting. So viruses are everywhere. That's the only takeaway here. The other takeaway is the almost unimaginable number of viruses. So there's a huge number of virus species, but if we're thinking about the actual physical number of organisms, it's not even worth thinking about. It's just such an unimaginably big number. But again, the takeaway from that isn't any specific number with 1,000 zeros on the end. The takeaway is that all these copies of the different viruses are slightly different. Even if we look at one infected cell where we have thousands of virus copies being pushed out, there's going to be tiny mutations that are littered throughout. So maybe some of the virus copies will be exactly the same as each other, but generally speaking, there's going to be huge variation. So this is a really, really, really powerful position for viruses to be in from an evolutionary perspective, because they can just generate this incredible genetic diversity very, very quickly, and that poses some very, very significant challenges for our immune system, because as I just said, our immune system has been evolving to prevent viral infections, so it's constantly been an arms race between our immune system and viruses. It's been tough. We see the immune system has evolved some very specific strategies to deal with this viral diversity that we will be talking about in future lectures. So we can say that viruses are basically everywhere, and that's basically true. but we can also think about some of the specific areas where viruses are, and think about the implications of viruses being there, particularly in the context of human infectious disease. So even though it's true that viruses are ubiquitous, specific viruses will be found in specific places. And I'm not just talking about geographical locations, I'm also talking about, for example, in specific reservoirs. So a reservoir is essentially a species that is typically infected by a particular virus. And often when we're talking about reservoirs, we're talking about viruses that infect these reservoirs and are then capable of jumping across and infecting humans. So if we're thinking about where viruses are, when we're talking about viruses that infect humans, one of the most important places we find these viruses is in reservoirs. One clear example is influenza virus. So influenza virus causes influenza, so it infects humans. However, it is maintained in reservoirs of avian species. So these are avian species reservoirs. So influenza infects various types of birds, avian species. The caridriforms and the enceriforms are the two groups. And really what we're talking about are gulls and waders and swans and geese and waterfowl. These are the types of birds that fit into these two groups. And influenza viruses infect these species routinely. Because influenza virus is capable of jumping to humans and then jumping back to birds, we consider these bird species as reservoirs of influenza virus, and they're actually a very, very problematic reservoir, and there's consequences to these birds being reservoirs. One of the consequences is our consideration of influenza virus as being ineradicable. The human viruses that we have managed to eradicate from the population, smallpox, really, we're only really talking about smallpox here. Smallpox we managed to completely get out of the natural order of things, in terms of there's no natural smallpox infections anymore because it's been eliminated, largely due to the implementation of the vaccine. I think the last case was in 1979 or 1980, and even that was an accident in a lab where there was some smallpox in the lab, and there's a whole big story about it that's very, very interesting. But the end result is there was an infection, and that was the last kind of smallpox case that I'm aware of, at least. One of the reasons why we were able to eradicate smallpox infection was because there's no animal reservoirs for smallpox. So if you treat or vaccinate everyone who has smallpox, so you prevent smallpox from spreading in the population anymore, then in theory you could completely get rid of smallpox, because it only infects humans. So if you deal with all infections in humans, then you're potentially eliminating the virus completely, and that's exactly what happened. Enough people got vaccinated against smallpox that it wasn't able to thrive anymore, it wasn't able to spread, and it was eliminated in that way. The problem for viruses like influenza that have these animal reservoirs, or reservoirs in species that aren't humans, is that even if you successfully vaccinate or treat, and by treat I mean control infection, every human being, and you stop the virus from spreading amongst humans, you still have these animal reservoirs. So you still have the risk that new versions of this virus will jump across from the animal reservoirs into humans. And that's what we see with bird flu and swine flu all the time. Swine flu, it's bird flu. It came from birds, then to swine, then to humans. All the bird flu pandemics we hear about, they're just influenza that's come from birds. Actually, technically, all influenza is bird flu, because all these new influenza strains we get, there's a new dominant strain each year, and this strain each year originates in birds. And that's why influenza is one of the diseases that you need to get vaccinated for every year if you want to be resistant, because it's a new strain jumping across from the animal reservoir every year. If we compare that to smallpox, completely different. We just vaccinated against the smallpox strain that exists in humans, and then we were able to protect enough people within the population that smallpox died out. So clearly these natural reservoirs for viruses are extremely important, and there's implications in terms of physically where these viruses are in the world. So there's specific regions where, so if you think about West Nile virus in subtropical regions, and even non-viral diseases like malaria, it's caused by a protozoa, but the principle is the same. The protozoa exists within Anopheles mosquitoes, like West Nile virus does. So it's kind of restricted in its geographical region because it's tied to this reservoir. So that's something to think about as well. Another consequence of these natural reservoirs is that we can potentially prevent infection or control infection by targeting these reservoirs. Someone said once that if we wanted to eradicate the flu, in the same way we eradicated smallpox, we'd have to kill every avian species on Earth. Obviously it's ridiculous and we're not going to be able to do that, and we wouldn't want to do that. So the consequence is we consider influenza ineradicable, so it can't be eradicated. For other infectious diseases, however, potentially we can target the reservoir, potentially we can eliminate the reservoir. So there's been a lot of efforts put into eliminating mosquitoes, for example, because a lot of infectious diseases, including infectious diseases caused by viruses, are spread by mosquitoes. Because again, if we go back to the West Nile virus example, the West Nile virus is transmitted by the mosquito. So if we can target the mosquito, reduce mosquito numbers, or eliminate mosquitoes, then we're eliminating the reservoir. All these reservoirs are also sometimes called the vector, by the way, because vector is how something gets from one place to another, and the mosquito in that example is how West Nile virus gets from a person to another person. So it's the vector. Yeah, so we can target these mosquitoes, and there's quite a lot of effort is being put into this and has been put into this. We'll talk about that in specific lectures. There's some really interesting ways in which researchers are targeting animal reservoirs and insect reservoirs, insect vectors, to prevent the spread of these infectious diseases. There's also endemic viruses. Endemic viruses are viruses that are just naturally in the population. You might have heard about the term endemic coronaviruses lately. There's a few coronaviruses that naturally infect humans. They've infected humans for a long time. They cause symptoms like the common cold. They're really not a big deal. And the reason is that we've evolved alongside them. So we've kind of evolved this relationship with them. That means we can tolerate infection. Obviously, these endemic viruses are very, very different to the viruses that jump across from animals to new viruses and things like that, and we'll talk about the reasons why, but we do need to understand that these endemic viruses exist. So these viruses that usually cause mild infection, that have usually evolved alongside us for a long period of time. When we're talking about geographical variations as well, as I was when I was talking about the insect vectors, we need to think about the context and like the real world. Obviously, there's things like income disparity and the disparities in socioeconomic status of different countries. These really impact infectious disease. The prevalence of infectious disease, the incidence of infectious disease, and the consequence of infectious disease at an individual level. Even just on the surface, if we just think about even at this most superficial level, if someone contracts infectious disease, do they have access to healthcare? The answer to that question varies dependent upon the country, dependent upon the economy of the country and the politics of the country. Is the country at war? Are there other events going on that impact access to healthcare. So straight away it should be clear that the impact of infectious disease is closely tied to these real world factors, that we kind of use this term socioeconomic to, and we'll explain that term a little bit more later. But the idea is we need to take this rounded approach to understanding infectious disease, and we need to understand that we're not just talking about things at a biological or physiological level. We need to understand the real world and the context. Okay, and just to summarize the different definitions of viruses that we've covered, because we named a few different definitions of viruses, or we can say we defined viruses in a few different ways. And I just want to run through what those were. So firstly, we said viruses are small, so they exist on a nanometer scale. Generally, they're smaller than bacteria, but not always. We also said that viruses are distinct from eukaryotic and prokaryotic life. So eukaryotic and prokaryotic life is cellular life. Sorry, it's cellular life. Eukaryotic life, eukaryotic cells. Prokaryotic life, prokaryotic cells. So these are two very, very different types of cell, but they are both still cellular. Viruses, meanwhile, are not cellular. Viruses are not composed of cells. They're not single cells. A virus is not a cell. A virus is acellular. When we're talking about a virus, we use the term virus particle, not virus cell. And the reason is a virus just doesn't meet the criteria to be classified as a cell. Doesn't have the organelles, doesn't have any of the structural features or functional features that define a cell. So instead, we define a virus as being acellular, A meaning not. So asymptomatic means not showing symptoms. Amoral, not having morals. I can't think of any other A's. There are some. Okay, so next, we define viruses as being composed of a nucleic acid genome wrapped in protein, sometimes wrapped in a lipid envelope. So this is the minimum composition that a virus can be. It is a nucleic acid genome, and again, that can be an RNA genome or a DNA genome. It can be single-stranded or double-stranded. It can come in lots of different specific forms, which we'll talk about later. But the key fact is it's a nucleic acid genome, so it's made of nucleic acids. And this genome is wrapped in protein. This protein constitutes the capsid, so it's a shell that encloses the nucleic acid genome, essentially. And this capsid is then surrounded by a lipid envelope. And this lipid envelope surrounds the capsid. It encapsulates the virus particle. Again, not all viruses have this lipid envelope, so this lipid envelope is not always present. When it is present, it is often studied with various proteins, and these proteins often play a role in the replication cycle, so in the entry of the virus into the host cell or the exit of the virus from the host cell. And again, we're going to talk about these things in a lot more detail later. For now, we just want to understand the definition, what constitutes a virus. Next up, we said viruses are obligate parasites. And again, what this term means is that to replicate, viruses are required to parasitize a host. In the relationship, the virus is the parasite, and a whole bunch of different species can be the host. But that relationship enables the virus to replicate, it enables it to produce copies of itself, it enables itself to produce progeny. So in the absence of this parasitism, viruses are not able to produce progeny. That's why we say viruses are obligate parasites, because they are obligated to engage in this relationship. We also define viruses as the causative agent of disease. And this is linked to obligate parasitism. We said viruses are obligated to parasitize a host. In a host-parasite relationship, the parasite gains something and the host loses. When viruses engage in a host-parasite relationship with us, the virus gains the ability to reproduce and produce progeny, as we've said, or to replicate. Whereas we, the host, we suffer some form of fitness loss usually, and often that fitness loss is experienced as disease. If we think about what the virus is actually doing, it's invading our cells, it's often destroying those cells. If it's not destroying those cells, it's seriously compromising those cells by robbing the cells of resources and things like that. So there's a cost associated with that, and that cost is often associated with disease. If we think about what's a good poliovirus, for example, poliovirus will enter into cells of the nervous system, motor neurons, I believe. And once poliovirus has entered into the motor neurons, it will replicate. As it's replicating and producing copies of itself, it causes cell death. So you eventually get cell death of all these motor neurons, and ultimately that leads to paralysis. And that's one of the main symptoms of polio. It's paralysis, and that's specifically caused by viral replication in these motor neurons. So they're a very clear example of how the virus is benefiting. It's gaining the ability to replicate, and the host, in this case us, is suffering, so is experiencing a fitness cost, and that's essentially what infectious disease is. Again, we also defined viruses as infectious agents, exactly the same concept as causative agents of disease. But when we say infectious agents, we're really emphasizing the point that they can move from person to person. So one of the characteristics of infectious disease is not just that it's caused by microorganisms, but also that it can be transmitted throughout a population, because these microorganisms are capable of moving from host to host to host. It's just not something you see with diabetes. It's just not something you see with most cancers, or genetic disorders, because there's no causative agent, there's no infectious agent that can move from person to person to person. Those diseases, those non-communicable diseases, are not caused by a specific infectious agent that can migrate throughout a population. So it's very important that we understand that these infectious diseases can spread, and the reason they can spread is because the causative microorganism, the causative agent, is capable of moving from host to host to host. Finally, we said viruses are everywhere. The term we use is ubiquitous, just all around. But we specifically thought about some of the particular areas that specific viruses can be, that are associated with, particular concern. So we talked about how specific animal reservoirs or insect vectors can form a place for these infectious agents to reside, and then these infectious agents are then capable of jumping from this reservoir into humans. Now we want to talk a little bit about virus structure. So we've had a little bit of a discussion about virus structure and kind of what composes a virus so far. What we want to talk now about is just a little bit more detail about what the structure of a virus looks like, and particularly we're going to focus on the capsid, because the capsid, and if we remember, this is the protein shell that surrounds the genome. The capsid is a very, very unique, very interesting structure that we do need to talk about in a bit of depth. So this is the structure that we've looked at so far. You'll see, just like we said earlier, we have the genome in the middle, then we have this genome surrounded by a protein capsid, and then this protein capsid in this example is surrounded by a lipid envelope, but this lipid envelope is not always present. It's just in some viruses. And while we're mentioning it, we refer to these viruses as enveloped viruses. So there's enveloped viruses and unenveloped viruses, and the enveloped viruses are called enveloped because they have this lipid envelope, whilst the unenveloped viruses don't. And there's consequences for this when we're talking about the viral replication cycle, but we'll talk about those later. The reason why the protein capsid is so interesting is because it's typically composed of protein subunits. However, these protein subunits are typically identical, so they're repeating, and they self-assemble into a structure. Viruses have a strong selective pressure placed on them to have small genomes, and we'll talk about why later, but for now we just need to understand there's a strong selective pressure for viruses to have small genomes, and one of the ways viruses can maintain their small genome is to evolve to use the exact same gene over and over and over again for the same thing, and that's what they've done with their capsid. So because the capsid is typically composed of either one protein repeated many times or just a few proteins repeated many times, that means a virus can potentially encode this entire protein structure, this entire capsid, with only one gene. And again, that enables the virus to maintain this very small genome. Again, we'll talk specifically about why that's useful, but if we think about the nature of viruses, it should be quite clear. Remember, these are parasites. They're trying to sneak into the cell and then replicate, ideally without being noticed by the immune system. The bigger the viral genome, the more likely it is to be noticed by the immune system. One of the things the immune system detects is viral genomes. So the viral genome is a very potent stimulant of the immune system. By maintaining a small genome, the virus increases its ability or improves its ability to evade the immune system. So we kind of use immunoevasion as a general term to refer to that. In terms of the shape of these capsid, and we'll often say things like virus shape, it's kind of true to say that, but when we're talking about virus shape, often what we're talking about is the capsid shape. So just bear that in mind when we're talking about the shapes we're talking about here. We're talking about the shape of the capsid, but we're kind of conflating that term with the shape of the virus overall. So we're talking about how these proteins assemble into a capsid, and what shape that capsid ends up as. One shape the protein capsid can exist as is a helical structure. And essentially what you have is the subunits assembling together in kind of a ring-like ongoing helical structure that has this rotational symmetry. Like if you look head on, like straight down, it just looks like a straw, and you can see it's composed of these identical subunits. What usually happens is the genome inside, so if you remember, we have the nucleic acid genome within the capsid. The genome is wrapped around the center of this helix. So again, it almost looks like DNA, but what it actually is a nucleic acid genome, which could be DNA, could be RNA, is kind of wrapped around the inside of this protein helix. One type of virus that has this structure is the tobacco mosaic virus. There's quite a few others as well. This is worth mentioning because when we think about viruses, we tend to think of the circular viruses, but they're not really circular. They're icosahedral, and we'll talk about those in a second. But viruses with this helical shape, they look long, they look thin, they don't look how we tend to imagine viruses in our head. So if you see viruses with this shape, usually what you're looking at is a helical virus rather than an icosahedral one. Next, we want to talk about the viruses that are icosahedral in shape. We have a diagram on the right here. This is the same shape, the same virus, looked at from 3 different angles, and we're going to talk about why we're looking at that now. Essentially, when we're talking about an icosahedron, we're talking about a shape that has 20 faces, and each of these faces is an equilateral triangle. So these faces should be exactly the same as each other. All should be equilateral triangles, and they're kind of jammed together into the shape of an icosahedron. The icosahedron looks roughly spherical. It's not spherical. As we can see here, we can see the sharp faces and edges, but it's roughly spherical. So that's why often when we're looking at viruses, they do tend to look spherical. Even the icosahedral viruses, they're not spherical, they're icosahedral. When this capsid is surrounded by a lipid envelope, which it sometimes is, we talked about, they look even more spherical because you can't see the edges, you can't see the faces, you can just see this lipid envelope, which is somewhat rounded. But really, at the viral core, we're looking at an icosahedral shape. So we can define the icosahedral shape in a few more ways. We defined it as being 20 equilateral triangles, which is very true. But when we have this icosahedral shape with 20 equilateral triangles, we have some other features that we can observe. Namely, we have 3 axes of symmetry. If we look at this diagram again, as I said, we're looking at the same shape, an icosahedral viral capsid from 3 different angles, and each of these angles is focused on a different axes of symmetry. So in figure A, we have twofold symmetry, which essentially means about this point that is highlighted, we can rotate the structure twice and see symmetry maintained. In figure B, we see we can make 3 rotations to a full rotation, and see that at each of those 3 rotations we have symmetry maintained. So at each of those rotation points we have one point of the triangle facing top. In figure C, we see the same, but we have 5-fold symmetry, so we can make 5 small rotations, and at each of these rotations we are maintaining symmetry. So 2-fold symmetry, 3-fold symmetry, and 5-fold symmetry, each about a different axes. These are the defining features of the icosahedral virus capsids. Okay, so we have said viruses have evolved to have these tight genomes, these small genomes. So it's beneficial for the virus to maintain a small genome, and one way it maintains a small genome is to evolve a capsid that is composed of repeating subunits. So the same protein made many times, and these repeating proteins assemble together into a capsid. Generally, the minimum you can create a capsid with is 60 subunits, and these 60 subunits are exactly the same as each other. So we have a capsid, this big complex structure, icosahedral structure with angles and faces, all created from a single gene, because it is made of 60 copies of the same protein. And the way this structure is organized at a subunit level is each of these equilateral triangles, so each of these faces is composed of 3 repeating subunits, so 3 copies of the exact same protein. We've said we have 20 equilateral triangles making up the icosahedron, so that means we have 20 triangles, and each of those triangles is composed of 3 protein copies. So in this whole icosahedral structure, we have 60 subunits. So we have 60 copies of the same protein, and that's how the virus has evolved to make these complex shapes out of a single gene. This example I'm talking about now is the simplest example. So this is the most simple capsid you can get. They can be more complicated than this. You can imagine where we have maybe 9 subunits per face. So per equilateral triangle, there's 9 subunits. We can imagine in that situation, it takes a lot more subunits to make up the icosahedron. But the principles are still the same, and we do see a lot of viruses that are able to create a capsid from as few as 60 subunits overall. Okay, we also have the complex viruses. So we talked about the helical viruses, we talked about the icosahedral viruses, now we have the complex viruses. These are strange. They're not shaped anything like the helical or the icosahedral capsids. They're so varied, it doesn't really make sense to go through all the different shapes we can have. All I really want to do is give you these examples here. Now these examples are bacteriophages, so these are specific viruses that have evolved to infect bacteria. So they will attach to the surface of bacterial cells and they will insert their genome into the bacterial cell. And then once the genome is in the bacterial cell, it will begin expressing, it will begin replicating, and essentially we have viral replication. As you can see, they don't look anything like helical viruses except for this long kind of stalk-like structure in some. They don't really look like icosahedral viruses except the head, so the top of the bacteriophage, and this is where the genome is stored. So this is something like an icosahedron, but not really again. So they don't really fit into any of the categories we have, and they also have these things that look like arms or legs that essentially help them to attach to the bacterial cell. So they don't look helical, they don't look icosahedral, and they're not helical and they're not icosahedral. So what we classify them as instead is complex. So we just lump all the other viruses together that have these non-standard shapes. We lump them together and just say these are the complex viruses. It's not a perfect system, but there are no perfect systems. We'll talk about more examples as we're going through the lectures. But you'll start to understand this stuff kind of as second nature. You'll just get to know, you'll recognize virus shapes as, this is icosahedral or this is a helical virus. You'll see just the long kind of worm-like structure or straw-like structure usually. And then you'll recognize bacteriophages as well. So basically all I really want you to do at this point is understand that these are the three main groups of structures that we have and that you should be looking out for. Now finally we have some information about viral replication. So I've talked a lot about the idea of viral replication and the idea that viruses need to enter into the host cells, and once in the host cells they can complete all the processes that constitute life. They can make copies of their genome, they can express their genome, and that all these processes allow the production of new virus particles. They allow the production of progeny. So all these processes put together can be considered the viral replication cycle. So I've alluded to the fact this replication cycle exists. What I want to do now is define it. So now look at the key stages of the viral replication cycle. This is just going to be really, really quickly because we're going to look at the replication cycle in detail later. We're going to look at the individual stages in real detail. The reason why we're going to do that is because this is what we target when we use antiviral drugs. When someone uses acyclovir for a herpes virus infection, when someone uses antiretroviral drugs to treat a HIV infection, or Tamiflu to control influenza infection. These antivirals are all targeting different stages of the viral replication cycle. So that's what we need to understand, the viral replication cycle, so that we can also understand how these drugs work and how we can develop new drugs. So this is a diagram of the viral replication cycle here, and what I'm going to do is break it into stages in a second, just to label everything, so it's kind of say what everything is. This kind of semicircular structure at the bottom is representative of the host cell. The particle that's approaching this structure is a HIV particle, so it's a virus particle. Inside the cell we can see the capsid of the virus particle. Then we can see multiple copies of this virus particle capsid, which means replication has happened. So we have new genomes made, we have new proteins made that assemble into the capsid structures. Then we have exit. So then we have these copies exiting the cell, and then we see them outside the cell, multiple copies. We've got about 6 here, but in real life we're looking at thousands and thousands produced by each infected cell. So we can try to look at the individual constituent steps of this replication cycle and understand the differences between these steps. So here we have labeled entry, replication and shedding, and these are the 3 key stages of the replication cycle. The virus entering into the host cell, the virus then replicating within the host cell, then the virus exiting the host cell. This process of exit is also sometimes called shedding. The idea is you're having fully formed virus particles leaving the cell, and they can then go and infect additional cells or infect additional host organisms. First off, entry. We need to understand this is different from entry into the organism. So we're talking about the virus physically entering into the host cell it infects, not entering into the host organism it infects. So for example, if you're by someone who has common cold and they're sneezing and you're breathing in the droplets of moisture that are full of virus particles, those virus particles are entering into your body. This is not what we're talking about here. This is not the stage of entry in the viral replication cycle. In that example, the stage of entry is when those virus particles then enter into your respiratory epithelial cells. It's not when they enter into the body, it's when they enter into the cell type they're infecting. Similar for HIV, it's not when the viral particles enter into the bloodstream, it's when the virus particles enter into the cell type they're going to infect. So for HIV, we're talking about CD4 cells, usually CD4 plus T cells. Again, yeah, and that's the type of entry we're talking about. Replication, really complicated step. We're going to talk about this at length later because a lot of drugs target this step. All these steps we're talking about are targeted by some drugs, but replication is particularly important as a drug target. Essentially what we're getting is copies of the viral genome made and also expression of the viral genome to create all the necessary proteins to make new virus particles. Once you have these copies of the viral genome made and also the viral proteins needed made to produce the full virus particle, these virus particles will then exit the cell where they can infect new hosts or just additional cells and things like that. And this process is called exit or sometimes shedding. It's important to note that this is when enveloped viruses acquire their envelope. So I said earlier that enveloped viruses acquire their envelope from the cell membrane of infected cells. What essentially happens is these viruses exit the cell, so during this process of shedding, and as they exit, they just steal a little bit of the cell membrane, and this becomes the lipid envelope. Okay, so I hope this was a useful lecture. Again, we're just laying down the foundational concepts. These concepts can be quite tricky, which is why we're going to talk about them in the tutorial later. But once you've grasped all these concepts, it gives you a really solid base upon which you can build your knowledge. So as we progress through the module, you've kind of got this good, solid foundational understanding, and you're not going to be held back. You're not going to feel limited by misunderstanding of what we actually mean by virus, what we actually mean by parasite and replication, and what a virus structure is actually like. So yeah, I hope it all made sense, and I'll see you in the tutorial. Thank you.